tryptophan operon or TRP operon. It's an anabolic operon. TRP operon is a repressible system that controls the metabolism of tryptophan, an amino acid. It works in a manner reverse to that of inducible operons. Inducible or catalytic operons are activated or stimulated when substrate to be catalyzed is present in the cell. On the other hand, repressible or anabolic operons get repressed or turned off when their end product accumulates beyond the needs of the cell. The TRP operon of E. coli is one of the best studied repressible systems. It consists of 5 structural genes, A, B, C, D, and E, which code for the production of enzymes that are involved in the synthesis of tryptophan, starting with corysmic acid. Also, it contains a promoter operator sequence, PO, promoter operator sequence, and a regulator gene, TRPR. The product of this regulator gene serves as a repressor. It is inactive by itself, so it will not recognize the operator sequence of TRP operon. It becomes active only when it combines with tryptophan. When the tryptophan buildup in the cell goes high, it binds with the repressor and activates it. When the tryptophan buildup in the cell goes high, it binds with the repressor and activates it. Thus, tryptophan serves as a co-repressor. The activated repressor is therefore the repressor-co-repressor complex. It soon recognizes the operator sequence, binds with it, and prevents the RNA polymerase mediated transcription of the TRP operon. This may be called repression of the operon. So it soon recognizes the operator sequence, binds with it, and prevents the RNA polymerase mediated transcription of TRP operon. This may be called the repression of the operon. Once the available cellular store of tryptophan is used up, tryptophan parts with repressor, which in turn detaches from the operator. As a result, transcription of the operon resumes and proceeds normally. This is called de-repression of the operon. Transcription continues until an excess of tryptophan is again formed. Now some tryptophan molecules bind with the repressor and the operon again gets repressed or shut off. This cyclic mechanism of alternate repression and de-repression of TRP operon ensures a balanced production of tryptophan as per the needs of the cell. However, this regulation is often modified by a second mechanism called attenuation, which regulates repressible operons. So we learned that tryptophan operon contain structural genes A, B, C, D, E, along with that promoter operator region PO, then this gene TRPE, corysmic acid is there, which is transformed into anthranylic acid with the help of this anthranilate synthetase enzyme, where glutamine is also involved. Along with that process, TRPE is active there. Then that anthranylic acid later transforms to phosphoribosyl anthranylic acid with the help of anthranyllate phosphoribosynthetase transferase. Anthranyllate phosphoribosyl transferase enzyme, TRPD, is active. Later that product which is formed earlier, like phosphoribosyl anthranylic acid, transforms to carboxyphenylamino 1 deoxyribulose phosphate. Carboxyphenylamino 1 deoxyribulose phosphate with the help of indole 3 glycerophosphate synthetase. TRPC is active there. After that, indole 3 glycerol phosphate is formed with the help of indole 3 glycerol phosphate synthetase enzyme. TRPC is active. Then TRPA and TRPB active. Later, tryptophan is formed from indole 3 glycerol phosphate with the help of tryptophan synthetase. So the genes of the TRP operon of E. coli and their role in the synthesis of tryptophan from choristmic acid, the end product will be tryptophan. We learned that. Later, attenuation process. Attenuation is a regulatory mechanism by which the transcription of bacterial operons which code for the enzymes of amino acid synthetase is coupled to specific control regions or attenuators near the 5' terminal of the mRNA. So when we try to learn about this, attenuation is a regulatory mechanism by which the transcription of bacterial operons which code for the enzymes of amino acid synthetase is coupled to specific control regions or attenuators. Attenuators are nothing else but control regions. Attenuators are control regions near the 5' terminal of mRNA. Transcription attenuation. Attenuation of repressible operons have been elucidated by Janowski and his team who worked with the TRP operon of E. coli. In this case, the control is exerted by attenuator region located between the operator and the first structural gene. Which one is the first structural gene? We learned about all those, but the arrangement is bit different. So first structural gene will be E. The mRNA transcribed from the attenuator sequence is called leader transcript. The base sequence of this mRNA reveals two surprising facts. The leader transcript has 4 regions whose base sequences are complementary to each other. There is a small gene called leader peptide gene whose bases from 27 to 68 code for a peptide gene. The 4 subregions with complementary base sequences can form 3 stem loop structures. Stem loop structures in the mRNA. Depending upon circumstances, regions 1, 2 and 3, 4 can form 2 stem loops, or the region 2 to 3 can form a single stem loop. When only a single stem loop is formed, the other 2 are preempted. The particular combination of stem loops determine whether transcription continues or not. The leader peptide gene codes for 14 amino acids, including 2 adjacent tryptophans. The codons for the adjacent tryptophans are significantly important in attenuation process. In attenuation, the cellular levels of tryptophan are very important. When there is excess tryptophan, the operator site will be available for RNA polymerase. This results in the transcription of attenuator site as soon as the 5' end of the mRNA for the leader peptide is transcribed. The ribosome attaches and initiates the translation of this mRNA. And depending on the cellular levels of tryptophan, 3 different outcomes are possible. If tryptophan concentration is high enough, translation will proceed down the leader peptide gene. The moving ribosome will overlap regions 1 and 2 of the leader transcript, allowing the formation of the stem loop 3 to 4. This stem loop structure is called terminator stem or attenuator stem. It causes the termination of transcription. If the tryptophan concentration is very low, the ribosome will have to wait for some time at the first tryptophan codon. This stalled ribosome will prevent the formation of the stem loop 2 to 3, which is often called preemptor stem. This will preclude the formation of 3-4 termination stem. So transcription will not be terminated and the whole operon will be transcribed and translated. As a result, tryptophan synthesis will continue, raising the cellular levels of tryptophan. Sometimes even though the cellular levels of tryptophan is very high, no ribosome will interfere with stem loop formation. So the stem loops 1 to 2 and 3 to 4 will be formed. This also will terminate transcription because of the presence of a 3-4 terminator stem. During this, ribosome is believed to be stalled at the 5' end of this TRP codon. This probably may happen when the cell is starved of non-tryptophan amino acids. it makes no sense to synthesize tryptophan when other amino acids are highly deficient in supply, hence cells can most efficiently raise the levels of other amino acids. So repressor site is there. Repressor co-repressor complex binds at the operator and prevents transcription. Repressed state is there. tRNA polymerase, repressor plus co-repressor tryptophan. Active repressor is there. Then E region binding will be happening. So DNA with repressor cooperator complex will be formed. Later, repressor dissociates from co-repressor and transcription is resumed. Derepressed state is very important, like inactive repressor. So transcription will go on and on. DNA without the repressor co-repressor complex. But when we look at this stem loop formations, you could see three kind of stem loops. Stem loop 3 to 4 ribosome, excess TRP termination, then TRP starved, no termination, like stem loop 2 to 3 ribosome, no termination but no translation, that means termination in case of third one, like stem loop 1 to 2 and 3 to 4. In both of these conditions, termination will be happening because there won't be translation, no translation is happening. Then TRAP control in some bacilli also, such as Bacillus subtilis, the TRP operon is controlled by attenuation. However, in this case, the secondary structure in the mRNA transcript is induced not by the binding of ribosome but by the binding of a protein called TRP RNA binding protein, TRAP. This protein first binds with tryptophan molecule and then attaches to the nascent mRNA transcript. This results in the formation of a terminator stem in the mRNA and thereby prevents translation. When tryptophan is not present in excess, TRAP will not bind to the mRNA. A pre-emptor stem, also called anti-terminator stem, will be formed instead of terminator stem so that transcription may continue.